Hello and welcome to the webinar about organic dry pea and lentil adaptation to South Carolina for plant-based protein production by Thavarajah and Tristan Lawrence of Clemson University. I'm Alice Formiga of eOrganic, and this webinar will last probably about 40 minutes or so, and then we'll have 30 minutes for questions. So today, it's my pleasure to welcome back Dr. Thavarajah, who is a professor of pulse quality and nutritional breeding at Clemson University. Dill leads the Pulse Biofortification and Organic Nutritional Breeding Program, and she also co-leads the Phenomics of the Feed the Future Innovation Lab for crop improvement at Cornell University. She is the 2022 FoodShot Global Award recipient for Pulse Crop Protein Biofortification. Today, I'd also like to welcome Tristan Lawrence, who will be joining us for the question and answer time. He is a master's student and he works as a project manager in Dill's lab. So welcome, Dill, and I'm going to hand over the screen control to you.
Thank you, Alice, and welcome, Tristan, and welcome everyone. It's my great pleasure here today to share some of our interesting work that we are doing at the Clemson University, South Carolina. What I'm going to talk about in next 30 minutes is about what we have been doing for last 3 years, and also especially last year, introducing a new crop, lentils, to South Carolina, as well as the work that we have been doing for dry peas. First, I like to thank my project team. We have a wonderful team, each and every individual, whether they have a PhD or not, they are a graduate student or undergraduate student, they have been extremely valuable and helping us to go through this journey, having the pulse breeding program here at the Clemson University. And I want to especially thank to Tristan Lawrence and Dr. Scheit, helping our program, and also many people like Dr. Borkwright helping bioinformatics, and many Nathan Johnson, all these people individually help in the program. Most importantly, we have a great partnership in the seed companies in the US as well as Canada. And also we are partnering with the ICADA Morocco and CSI Spain. Also, I like to thank the Good Food Institute and FoodShot, Global FoodShot, and also Clearwater Group, and also the Upper North Carolina Farm Group, Carolina Farm Association. And they have been wonderful to us as well. And my advisory board, I like to thank you for them, their guidance and advice on our program. Today, I'm going to talk about a little bit about the plant-based protein market and how dry pea and lentils going to fit in there. And then what are the stakeholder challenges in South Carolina? And this talk is basically entitled to South Carolina, but it helps everybody around the states, nationally as well as internationally. And we are going to share some of our field results from last 3 years as well as the last year. So let's talk about, or let's figure it out what's going on with the US plant-based protein market. So right now, in 2018, the global protein market, the US market, is about $4.8 billion. But it is now in 2021, it's $7.4 billion. If you look at the USDA data or the Good Food Institute annual report, you can see how the data that they found out around the US and the global market, how this increased. It's about 10% increase. So their projection for 2025 is $10 billion. So what this increase? It is basically because of your favorite Burger King choice, Impossible Waffle, and the Beyond Burger, and also the range of organic proteins that in the market. But what is the major ingredient in these proteins? Most of these ingredients are soy protein or pulse protein. But soy protein are currently getting some bad press because of their allergen effect and some health implications, especially for women, it's conflicting. There is no 100% evidence on that, but there are publications are coming back. For example, I'm allergic to soy, so I can't actually depend on soy. Therefore, pulses, especially in the organic market, this is a niche market for organic production to increase the protein production for the pulse growers. When you look at the dynamic purchasing dynamics in the US, and you can see this also data collected from the Good Food Institute for 2021 to 22. And the most popular, I am sure most of you know, most popular thing is like the plant-based milk. And so pulse industry has a great opportunity to make some sort of a plant-based milk, plant-based protein products, protein powder for the burgers. So when you look at the American household, 6 out of 10 households purchase plant-based food. And the milk is the number one. So most people coming out of from the dairy products to the plant-based milk or plant-based burgers, yogurt, meat, and range of food products in the market. If you walk in the frozen section or even some in the fresh section, that you can find quite a bit of plant-based protein products. So here is the new opportunity for pulse growers and the pulse processors having pulses as a major ingredient for protein production. Let's look at about dry pea and lentils, what they are and what is the nutrition quality compared to the cereals. For South Carolina, organic pulse crops, we use it as a winter catch crop. So mostly farmers grow these crops in January and they wanted to harvest it in May or early June before the heat come in like the hot summer. Why pulses are very important? Because they fix their own nitrogen and also they provide enough phosphorus and the crop rotation and also the beneficial bacteria back into the soil. It's improved the soil health. So you don't have to add any nitrogen fertilizer into the soil because it fixes its own nitrogen. So that's the part of the carbon benefit and the nitrogen benefit and the environment benefit. But there is a huge benefit for the consumer. That is the nutrition benefit. That's my whole my career dedicated for the nutritional side of the pulse crops. So pulses are rich in protein, 20 to 25%, and rich in prebiotic carbohydrates, about 10 to 20 grams. These are the food for the gut microbiome. So if you wanted to have a healthy gut or you want to reduce your weight, pulses is your choice. Low digestible carbohydrate and also micronutrient, gluten-free, allergen-free, because it's organic as well. So therefore, there's no chemicals added. And our research on last 10, 12 years that in my whole career, we did human studies, we did animal studies. We have proven that having pulse-based diet would reduce the obesity-related health complications. Also, having pulses would increase the iron intake, selenium intake, as well as the selenium and the zinc intake. So when you consider compared to the other crops like rice and wheat, this is a very good source of protein and low digestible carbohydrate. You don't have to worry about this carbohydrate. So that's the nutrition side. So what is the stakeholder needs? Here we divided stakeholders into our producers as well as the food processors and the consumers. So the production, we have a major issue in South Carolina because we do not have organic adapted cultivars. Current cultivars in production, which is produced by, which is developed by Crop Development Center in Canada or Alberta, Canada, or in Washington, or North Dakota, or Montana. So all those cultivars are mostly adapted to the conventional farming system. So we are testing their cultivars in our organic production. Sometimes some cultivars do better, some are not, because these cultivars are not adapted to the low input system. And then the misconception of the nutritional quality. Most people think organic produce has less protein, and some people think they are very nutritious. These are all misconceptions. And actually, organic crops are growing in the low nitrogen input, and so therefore they can have less protein, but they are chemically free. So therefore you can say it is healthy, but we have to analyze and we have to prove the nutritional quality, which is a misconception that saying that is very high nutritious compared to the conventional or vice versa. And the management issues we have, especially weed control issues we have, and then the duration of the maturity, flowering time, and everything is very difficult when you grow these spring adapted cultivars in the winter time. And not only that, what are the food processing issues? Pulses has a high amount of protein, but it is not a balanced protein because some of the essential amino acids are very, very low to moderate. I wouldn't say there's no, but there is these essential amino acids, but they are in low concentration. And the next problem is most plant-based food are not highly digestible compared to like animal protein. So it's a problem there in digestibility. So food matrix as a whole has a need to be studied and well understood when we talk about protein and the protein quality. Not only that, these peas and lentils, food processors use them as ingredients and they extract the protein out of these. And once you extract it, you dry them. And then you actually concentrate all the chemicals and toxins and pesticides. So are those helpful for our human health? So it's a question. So these all are questions about the protein stability, protein function, protein structure. When you add your protein into your morning smoothie, you don't need to be separated into two sections. You need to have a nice, very smoothie had to be very nice, homogenized, mixed. So these are problems that having the protein powders or use it as burgers. So you need to have a proper digestibility and proper nutrition quality. So these are the challenges we are trying to address from our project. And one of the major things that we are doing, addressing in the pulses is the protein quality. Most of our breeders are looking at the protein content, and the protein content is about 20 to 25%. But we know amino acid balance is different because protein has pulse protein has low sulfur containing amino acids compared to the cereals. But then cereals has higher sulfur containing amino acid. So that's why we usually balance the pulse protein with the cereal protein, get the balanced amino acid profile. But then when you think about the protein quality measurement, something we do it in our lab, if you talk to one of my scientists like Nathan, who graduated very recently, to run the amino acid profile, it will take, to run one batch, it will take minimum 2 weeks. But per sample cost would be $100 per sample just to get the amino acid profile. But if you just get the nitrogen content, even that will cost $10. So these are very difficulties in the breeding programs that you can measure protein content using NIR. NIR technology may be like it will cost your sample $5. But then what about the protein quality? So these are issues in the breeding programs that you not only measure protein content, you need to measure protein quality. So these are the things we try to address in this project. With that, our goal of this project, this is a 5-year project funded by the NIFA OREI. And our long-term goal is to develop the lentils and dry pea cultivars suitable for organic protein production. We are trying to develop high quality proteins, and we wanted to have a better digestibility, color, texture, aroma, flavor, and better adaptation to protein isolation and blending. What that mean by we wanted to preserve our protein structure when we have our smoothies or our food products. We wanted to have a nice blending. So all of these sections we're going to address in 5 years, and we are confident we have quite good results. In the last couple of years, we have been working on this project. I'm going to share the last year and last 3 years data for peas and the lentils. And our on-farm location, all these breeding trials we have been doing in on-farm locations in South Carolina, 2 locations close to the upstate, Cherry Farm and our student organic farm. And then the WP Rolls and Sons, the biggest farm that we have been working with them, they are the largest scale growers in the United States, and their organic farm is USDA certified. All these farms are USDA certified except the Living Web in the North Carolina, which is in the transition. And the Lomax farm also in North Carolina, also USDA certified land. Most of our breeding trials we have been doing for last 4 years at the Columbia, at the Pea, WP Rose and Son, which is a standalone soil. Here are the dry pea conditions that we have been growing the advanced lines from 2019 to 2022. We did not do field work much in 21 because of the COVID. And these experiments were happened in the USDA certified farms. And you can see in this picture, this is Pea location. You can see Tristan is taking some data. What we did was we planted around late January when the soil hit about 6 to 10 centigrade, a little bit of warming up, not before that, not after that. So the planting window for South Carolina is the last week of January or first week of February for these peas and lentils both. Clemson location is more warmer, and then also we have clay loam soil. And the Pea location, we had sandy loam, and which is very nice. We did not have much of the disease pressure because of the soil that we have. And the lentils, we had 3 locations in the lentils, actually Lomax Farm and Asheville and the Pea. And you can see here, Lomax Farm is in North Carolina, which is very, very cold. So this coldness in the January might be a problem, which I'm going to talk about a little bit. And also management is critical in this kind of soil. So these pictures are from Pea location. You can see the lentils are growing very nice with that sandy soil. And also we had about 50 square feet plots. And we did the soil sampling. And no irrigation was provided. We add the rhizobium inoculum. That's all we did. We didn't add any fertilizer or anything because this is the first year we introducing the crop. We wanted to just see how it was doing. And most important thing that I wanted to emphasize on the organic agriculture is the field management. So the before planting as well as the after planting. Before planting, it is very critical. So you have a fall ground cover, or you have a cover crop. So you should be discarrow it or crimp it before you start planting in the January. So what we did, this is the management schedule from the Rolls farm. This is how the Tristan can talk more about the management. And this is what the organic farm manager does every year for their farm management. And they actually don't have much weed at all in the organic trials. So we did a lot of harrowing, and then we plow it 3 weeks before, and then we cultivate 2 weeks before. And then whatever the soil amendments that you use based on the soil assessment. And this trial, we did not add anything because these soils were originally low in nitrogen, but they have moderate to high amount of phosphorus. We did not add anything because we were doing trials. And even the farm manager cultivate the land 1 day before the planting. And also the after planting was very critical to control the weeds. We usually after planting, it's take 5 weeks, about 30 to 40 days to canopy closure. So about 5 to 6 weeks. And that is the very critical time that we have to manage the weeds. So if we have weeds before the canopy closure, we will going to lose quite a bit of our experiment sites. So therefore, we did cultivate actually 1 to 4 times to control the weeds. So you can see that there's no weeds, and this is how we did in the organic system. And the soils were good. We did not see much of the nutrient deficiencies in the pillion location. And these are the pictures from the POP diversity panel here at the Clemson in my left-hand corner. And also this is also cherry farm in Clemson. That's our advanced trials that we were looking at. Also, we had some diversity panel growing here in the cherry farm. So you can see that we don't have much of the weed if we had that program that we have been cultivating this land every 4 weeks, every week before canopy closure. And this is the advanced lentil trials at the rows. And you can see here, this is the, you can't see much of the weeds there. And these were very nicely managed plots. And same here, this is our lentil diversity mapping population that we grow at the pillion. You can see quite a bit of the different diversity, and it was a very successful last year, our panel. So this is 2 PhD projects that working on this. Also, we are using some of these accessions to develop short duration cultivars for South Carolina. And these are the advanced trials in the rows. And then I want to talk about a little bit about the North Carolina trial. So this is how the North Carolina trial looks like in Lomax. It was unexpected. We had wheat root rot, and it's looked like more like a disease nursery. And it's quickly infected. And what happened was the early stage, we did not quite manage the weeds, and then later they start managing the weeds. But at the early stage, this is so important. Their soil is much wet compared to the rows, and so the diseases can happen very, very quickly. So we have been working with the farm manager in the Lomax farm, also Mark Dembski, and he has been so helpful on this project, and they were helping us quite a bit to control this disease. And then he got samples and he sent it to Utah. She's Dr. Utah from Montana State University Diagnostic Lab. And what her diagnosis was, and she thought it was the root rot as well as the Fusarium feces. And then when you look at that trial, it's just like a disease nursery. And then looking at the varieties, and one of the Dr. Vandenberg's varieties were actually resistant, which is very nice. And everything else was destroyed except the one variety. And Dr. Utah's recommendation was that first of all, we need to have a clean field, and maybe delaying planting. That is one of the things, because North Carolina soils are quite cold in January. Maybe we need to change the planting date, which we are talking about it this year. And then the seed treatment, we have to find a USDA certified seed treatment to control the root rot, as well as the number one thing is the weed management. And also her recommendation was avoid 5 to 10 years of the lentil in that field. So we are not going the same location, we are going to a different location. So what my point here is, the field management is quite important in the organic production. So with that, because of all of these, this is part of Tristan Lawrence's master's thesis, we decided let's talk about what happened with the planting date. So we have been doing this for 2 years with the late, early cultivars, with the control and the borders. And we have fall planting, actually 3 fall planting and 1 winter planting. And here is the results. So what happened when you do the fall planting in South Carolina is fall is actual, really, you feel the fall. And also like this morning, you feel really, really cold. So you have cycles, hot days, cold days. So what happened when you plant in the fall, it's the pulses start growing very quickly, increase the biomass, and then we get the diseases and they destroy. But if you plant in January with the cold weather, it's persistent. So here is the yield data that the selected cultivars. You can see the things that we planted in January actually work much better compared to the fall planting. Basically, in the 2022, it's wiped out. Everything was wiped out in the fall. So the January planting is very important for the South Carolina, but a little bit later planting would be okay for North Carolina. So a little bit about the results. So these cultivars we tested was from conventional lines, and we see the data range from 700 to 2,500 kilograms per hectare. Some varieties like ACC Carver from the Canadian program is wonderful, but we had to watch for the organic system, and because of the duration of our growing season. And if you wanted to grow something for protein, CDC Striker, which is also from the Canadian program, Dr. Wackington developed that. That one is wonderful protein quality for the organic system. So if you are a grower, you wanted to have dry peas right away in this winter, you can plant one of these cultivars like AEC Carver or CDC Striker. And actually these cultivars perform really, really well. And all this data we put in our website. Also, this is published in the PLOS One journal. And these cultivars are exact same as the numbers we've seen for nutritional qualities like protein, minerals, and the prebiotic carbohydrates, same as the conventional numbers. And if you want to read more details, you can go on this paper and look at it. But I would recommend the AAC Carver as the most adapted cultivar for the organic system here for South Carolina or North Carolina. And let's go on to the protein quality. So that was the basic parameter, yield and the protein content. This is what the growers are quite interested on. And this is what the processors are interested on. So how much protein actually would be digested into the human body. So what we did was we got that protein and we look at the liberated total amino acids. And then what we found was it is about 11 to 22 grams per 100 gram, even though protein content can be 27 grams per 100 gram, but about the total amino acid content, the liberated amount would be 22, very less compared to the total. So that's why you might not be able to increase the total protein content, but you might be able to work with the amino acid content to change the protein quality or increase the protein quality or the digestibility. And the problem with these amino acids, they have very low heritability estimates, and also these are quantitative traits. So breeding for these kind of things are really difficult. And that's the quality of the protein. And you can look at, and this data also will be published in PLOS One very soon. And you can see here is the amino acid profiles. Also, we calculated the recommended daily intake for the healthy young male or women. And you can see quite a bit of amino acids like sulfur-containing amino acid cysteine is about 15 to 85 recommended daily allowance. So which is very good. So these proteins provide enough amount of essential amino acids and amount of liberated total amino acid change from these cultivars that we tested. Also, we looked at the in vitro digestibility, and digestibility was ranging from 83 to 95%, which is very, very good. But we still have a lot of work to do because these are adapted to the commercial conventional cultivation system. So when you adapt it to the organic system, these might change more. So we need to do more studies on this work. And this is our germplasm, like when you look at it and how the distribution of the cysteine content, methionine content, in vitro digestibility, and the total amino acid. These are the traits that we're looking at in our breeding program to increase the nutritional content or the protein digestibility. Now let's move into the protein quality. So once you have these cultivars as organic cultivars, we extracted out the protein. So when you take the protein extracted out from field protein, These proteins are about 74% protein in that extracted protein. Also, it contains the other prebiotic carbohydrate, carotenoids, folate, as well as the minerals. So there is the processing techniques, and our lab holds the patent for it. And the patent will be published end of the. It is publicly available right now, but it will be published very soon. It is submitted. So this protein has the organic protein yield about 70 to 90%, and also using this extraction methods, it has a better taste and a better flavor than the control. So when we compare, these controls are from the market protein available in the Walmart or in one of the big stores, and we compare that with organic field, pea protein, as well as the sorghum protein. And when we combine field pea protein with the sorghum protein, it becomes a balanced, healthy diet. And also, I wanted to make this point because the extraction procedure that we developed in our lab, which is patent, but it's changed with the cultivars. So these are the modern cultivars that we have in the market, and each cultivar we use with our method. And then what we found was the protein efficiency or the protein extraction is different. So there is a genetic component into the protein isolation as well. And these are the chickpea protein and the pea protein, and then we use this in the food product development in our lab. We make bakery products and we make milk out of it, and then we have smoothies, and we make quite a bit of range of food products in our lab. There is a team of people working on it on the food processing side, students, and actually Dr. Thavarajah leads that. And this is the 2022 lentil variety trials, that the first time we tried lentils in South Carolina. But when you look at the total protein and total starch it's a little low, but it's in the good side. But the protein is quite high. We are very impressed, but we are not very impressed with the yield. This is not because of the problem with the variety. It's a problem with our harvesting as well. The problem is when you put these conventional varieties in the organic field, they are not grow exactly the same as the conventional field because the nutritional status of the soil is quite different. So therefore, the plant height architecture is a problem. So they are shorter than the regular size. So then when we combine them, we lose quite a bit. So in our combine, we lost quite a bit of yield. So therefore, this yield, actually, we think that about 40% of the yield we lost it because our combine could not catch everything. So if we actually measure it by hand, we should be able to get the exact amounts of the yield. So this is the first year. We also introducing a new crop, and we learned the difficulties. So our breeding pipeline, about that, our objectives are to develop dry pea cultivars adapted to low input organic on farms with increased nutritional quality. So this is Dr. Shive. He comes and he has been tremendous help on our program with Tristan. And so right now these are advanced breeding lines that we are doing some seed increase, and we are trying to test these in the field next year, 2023, in a couple of months. And in end of January, we are hoping to plant these in the field. And our traits are yield, plant architecture, short duration, because we are looking for 90-95 day cultivar, and the protein quality, digestibility, prebiotic carbohydrate and mineral, long range of traits, which are quantitative traits, which are very hard. But we have some tools that we developed that we could increase the phenotyping fast. Also, we are moving to a genomic selection, and we have the breed base for breeding management and analysis of software, which is curated by Dr. Johnson and Dr. Portwright, and Dr. Lucas Portwright lead our bioinformatics pipeline. And target dry pea organic cultivar release is 2025. And the closing remarks, so this is Sonia and Tristan working in the breeding program, and they are doing a lot of process, and these are advanced breeding lines, and these are our lentils. So organic pulse production is quite feasible in South Carolina. But the problem is we need to watch for the field management, which is very, very critical before and after planting. And also we need to watch for the rhizobium and watch for the previous cover crop. And there is not much of the diseases we saw in our last couple of years, but the planting day is very, very important to avoid the root rot or other diseases. And if you have a disease issues, that you could contact the Clemson University Diagnostic Lab. Also, Montana State University is fantastic to do all this diagnostics for the pulse crops. And we recommend AAC cover as the best adapted cultivar for South Carolina. And if you are working for protein, and CDC Striker is the one that we see continuously is the better protein that providing for us. And these seeds are currently available. You have 2 more months, and you could get these seeds from Meridian or Pulse USA from North Dakota. And Clemson University will be releasing organic cultivars very soon. And also you all can check our media release and our website and the social media. And if you're around Clemson area or in South Carolina, please join with our first field day, which is scheduled for April 13, 2023 at the WP Halls Farm. And this is our social media outlet, and that is my wonderful group. And I am urging you guys to eat lentils and try lentils. If you don't know how to cook lentils, check our website. We will be having lots of recipes. And also when you go to a restaurant, just order pulse dish just to try if you don't know how to cook it. But these are very easy cooking. You could make hummus, or you could make any kind of salad. And these are the dishes that we develop in our program. And any update, you can go onto the website and the YouTube channel, and we have Twitter. Also, you can join our Going Organic webinar series every month. Actually, we have next week a webinar. Dr. George Vandermark is talking about chickpea breeding and management. And with that, this is our funding, generous funders that help our programs, especially NEPA, OREI. The GFI funding has been excellent for our protein pattern work, and also the FoodShot Global South Carolina Department of Agriculture, USDA ARS Washington Chickpea Breeding Program. I'd like to thank Dr. George Vandermark and also Clemson University for supporting my program and helping with everything. So this is all I have, and thank you all.
Okay, thank you, Dill. You know, this is such an interesting and complex project. It must be fun to work on because there's so many different parts to it. I'd like to encourage everybody to type in questions. It's a great opportunity to ask Dill any questions you have about any of this directly. First one, could you please remind us if species-specific rhizobia inoculations were used for all of the field trials?
No, we didn't. We actually used the rhizobium that's specifically for peas and lentils. It's not the rhizobium species specific. So we bought it from actually commercial ones because we want to do this available to farmers. So Tristan can give the more details on where you bought, but these had to be USDA certified, organically certified rhizobium specifications for dry peas and lentils. Is that correct, Tristan?
Correct. Yep. So we used active pulse granular mycorrhizal inoculant with our peas. And it was applied at about 4 pounds per acre. And if you can apply it in the furrow, that's preferred. So you don't have to work it in, or we applied it with the seed if we didn't have that capability. But yes, I mean, for the peas and lentils, and there is specific inoculants, but those are the ones we used because they're organic.
Yeah, when I was a student, I used to make my own rhizobium strain, but we can't do that here, like developing the exact specific strain. So what we did was to combine commercial, because we have to be helpful to the growers.
Okay. Here's a question about whether we can adopt what you do or your approach in South Carolina in other parts of the world, for example, Asia.
Absolutely, we can do that because in here in US, it is very strictly for the USDA organic certification. So we do not add anything to jeopardize our certificate for our growers. So we make sure our seeds are USDA certified, no chemicals added, and very low input. But when you think about Asia, I'm from Asia, so I'm originally from Sri Lanka. So I know it is already organic. There is no certificate, but the land is you can keep as a you can have a crop rotation, and that is organic. So you can incorporate other legumes into your cropping season. You change it, and that will break the disease cycle. You can, so that way you will add very low amount of fertilizer for the subsequent crop. And also you can manage, like if you have rice and peas. So you have the rice on the rainy season, and then once the dry season comes, you can have a short season legume. So ICARDA, International Center for Dryland Agriculture, has a lot of information on how you can do this. Actually, that's why we did our first webinar with Dr. Shiv Kumar and finding out how we can adopt this kind of agriculture into the organic agriculture into Asia. So if you want more details, I would recommend you come to our YouTube channel and check Dr. Shiv's recommendation to have this kind of projects in Asia.
Okay,
and
great.
It's very, very easy to do that. And you can reduce the synthetic fertilizer. But the problem, don't think that you can do organic overnight. This is the long-term process. Because I have been reading some news from different countries in Asia. They try to cut the fertilizer, nitrogen fertilizer overnight. Please don't do that. You go step by step, because organic is a low input, slow system. So you adapted into the system very slowly. Even here, we have 3 years for the transition. So you need to read a little bit. You just cut down your pesticide, bringing your sustainable approaches into the system. So think this as a system.
Thanks. Let's see. Another question is, why do you think protein content can have such low heritability estimates?
Because protein content controlled by so many different genes. Because when you think about amino acid, how many essential amino acids human required for the body, but in the plant there is many other amino acids. So each amino acid has a very different way of controlling the biochemistry in the plant. So these amino acids act as the families. So these families were controlled by so many number of different genes. So that's why it is very, very hard. Also, do not forget, always protein content and the yield has an inverse relationship. So when you increase your yield, your protein content anyways go down. It's a dilution effect. So this is very hard to increase the protein content in the genetically, and that's why all those breeders use very diverse populations, bring different diversity, and that's why we work with ICARDA, and that's why we work with Spain to bring this different diversity into our breeding program and see whether we could find higher protein content at the same time the protein quality. I'm not much worried about protein content anymore. If you get the decent amount of protein, like 22, 23 for your advanced breeding lines, and the grower get the proper price, I am more worried about, is this protein going to be digestible in the human side? So it's a good question. We are learning.
Let's see. One of your goals is to produce pulses for protein isolation for use in smoothies, for example. Are these cultivars equally suited for cooking with directly, or would ones bred for that purpose be better?
That's a great question. This is exactly what we are seeing in the commercial cultivars right now. We have isolation process. This is the trend, right? Right now, everybody love to eat the plant-based milk, plant-based organic, not plant-based burgers or protein powder. We see a difference between isolation and the whole seed. As a person who eats pulses quite a bit, because this is my main protein source, I recommend the whole food, because whole seed will give you everything else, right? You don't discard the carbohydrates and things like that. But when you do this isolation, actually some of the carbohydrate you're going to lose, some of the carotenoids you're going to lose it. And yes, some of the cultivars are better for this isolation process than the others. So that's why at the beginning I was saying that growers has different challenges and the processes has a different challenges. But if you are doing a niche market and someone actually doing very highly specialized protein for very high-end food products, we will have that kind of cafe type cultivars developed for that purposes.
Really is amazing how diverse different pulses are. Because I mean, even with lentils, I know that there are certain types of lentils that are commonly sold in supermarkets, but then there's some that are in the bulk section of food co-ops and things like that. And there's a huge amount of variation in terms of cooking time and the taste and the qualities of these different types of lentils. So, I'd be really interested to just sort of see a publication all about different types of lentil cultivars and what their cooking properties are. I don't know if you're planning to just kind of publish something about all the different cultivars that you've explored and their food functionality.
That's the plan. In my group, there's a team of people leading the food processing side. That's our plan. But there are more on the processing side. There are patents are coming because it's about how we take them out from the seed and how much you're going to leave the nutrition in that isolate. So it's very unique, and you can add so many chemicals, or you can add very less amount of chemicals. At the end of the project, we will be summarizing all the protocols that we do, and we are hoping to put that in our website so that people can use all this information for generating new cultivars as well as in the processing market.
Great. Okay, we have a question about what the sulfur status is in the experimental sites.
Trista, do you know that?
Do not. No, I would have to look at the soil samples.
I don't think we know that, right?
Okay, that's fine.
Because we never test sulfur, it's NPK.
Okay. Let's see. What are the seed components that impact digestibility, and how do you evaluate and select for better digestibility?
Well, the digestibility depends on the food matrix. So there are protein inhibitors and anti-nutrient factors in the pulses. So one of the things that we are actually interested is phytic acid. But in my previous work, when I worked with the peas and lentils in Crop Development Center, and the germplasm that we work actually has very low amount of phytate. So I'm not actually worried about much of the phytic acid. So in our breeding program here at the Clemson, we try to keep the target as 100 to 300 milligrams per 100 gram of phytate as the baseline. So if you have low amount of phytates or moderate amount of phytate, I'm not saying very low, because if you reduce the phytate, then plant will not grow because we will not have enough phosphorus for the next generation. So phytate is the one of the component that to play with the digestibility. And also there are other things called trypsin inhibitors and some other anti-nutrient factors like tannins. So most of these peas and lentils, they have very low amount of dose. And we are still in a process of looking at what are the individual factors that are going to be affected on the digestibility. And how to measure the digestibility? you can measure the digestibility by using animal model. The easiest model is the chicken model that you could do it in a, if you talk to Dr. Jerry Combs, he would say you could do this in 2 months because it's chickens. But we don't do that in the breeding program because we wanted to do quick and easy method. So we use the enzyme assay digestibility method, which is available in the mega enzyme. But it take also time. So one of the things in one of our PhD student and is developing the upcoming new PhD student. We are developing a high throughput phenotyping pipeline that when we get our seed, whether we could measure the digestibility using a technology similar to, it's not NIR, it's called FTIR. We are still in the progress of measuring the dry seed digestibility. Other than that, that will be available for the breeders once we develop the protocols. But the only way right now, you have to use the enzyme digestion method or the chicken model.
Okay, did you find the heritability values for protein content surprising? As legumes with the ability to fix nitrogen, should we expect protein content to be more stable and consistent than cereal crops?
Well, our numbers were within the range of the literature, but I have to say this. So this data is more like the heritability calculations were, I have some sort of a question in my head, because usually when we do the heritability estimates, we do several years, several locations, and with several reps. But this study were done only 3 years with certain number of germplasm. So that's maybe why our numbers are a little low. But with the breeding populations, we are trying to make a better estimates. But I don't see a connection that having a nitrogen fixation which increase the protein content, which have a higher heritability. I think we are talking about 3 different concepts. So I don't have a correct answer for that because I don't have supporting data to provide that. Our estimates are low. Only thing I know, our estimates are low. That's why we are working very hard to bring the genetic diversity into our breeding program.
Okay, yeah, that may answer the last question that we have here, which says, with relatively low heritability of protein concentration, what breeding strategy do you use in your program?
Yeah, first of all, number one strategy is we need to increase the diversity. That's why we have been creating this mapping population, which will be going out to the field next year, and which has about 400 accessions that we collected just for protein from different parts of the world. That's why we have this partnership with ICARDA and Spain. And I think with that, we might be able to have a better diversity in the protein content. And also we are developing more mapping populations with Sonia's project for her PhD. And I think we will have quite a bit of answers for that near future. Also, we can increase the diversity. That's not a problem. We can make connections with all the breeders around the world, and somehow we can make MTAs and we can exchange material. But how do we do these thousands of samples for the phenotyping? That is the biggest question that we're going to answer in this project. That's where we developed this pipeline, whether we would go on the genomic route or we would go in the phenomic route, actually having a quick and easy tool like FTIR, which we published a recent paper from Nathan's PhD thesis, whether we could use one seed actually to treat all of these samples. I haven't shown that data. This summer we had about 10,000 samples altogether, but in the breeding program samples, it took us a while to get the protein numbers around 6 weeks, but it is still faster than running through the LECO or the digestibility. So this new platform will be coming. So what you need is you need the genetic diversity. You need better tools to quantify these amino acids or the protein digestibility, whether you use the genomic production tools or phenomic production tools. Those are the strategies that I can think of. Also funding and how much money that you have in your program. So those are my strategies right now I have.
Okay, if anybody has any last burning questions, we have another few minutes, so feel free to type them into the Q&A box. Meanwhile, I just want to remind everyone that we will be posting the PDF of the slides as well as the resources that Dill mentioned. So, for example, the social media sites associated with the project. We will link those to the archived webinar on the eOrganic website. So the easiest way to find it is just to Google webinars by eOrganic, and then you'll find the list of all of our webinars along with the associated resources. And we will have those up within the coming week. I just want to thank you so much, Dill and Tristan, for this very interesting presentation, and we're looking forward to having you back once you have more results in the project. So thanks everyone for the questions, and thank you, Dill and Tristan and everyone for joining us today.
Thank you so much.
Thank you.